The digestive system is a complex network of organs and tissues responsible for the breakdown of food, absorption of nutrients, and elimination of waste. In vertebrates, this system comprises the alimentary canal and accessory organs, each playing a pivotal role in ensuring efficient digestion and nutrient assimilation. Alimentary canal components. Mouth. Digestion initiates in the oral cavity, where mechanical breakdown of food occurs through mastication. Salivary glands secrete saliva containing enzymes like amylase, which commence the chemical digestion of carbohydrates. Esophagus. A muscular tube that conveys food from the mouth to the stomach via coordinated contractions known as peristalsis. Stomach. This sac-like organ serves as a reservoir for ingested food, mixing it with gastric juices rich in hydrochloric acid and enzymes such as pepsin, facilitating protein digestion. Small intestine, comprising the duodenum, jejunum, and ileum. The small intestine is the primary site for nutrient absorption. Here, chyme from the stomach mixes with bile from the liver and pancreatic enzymes, enabling the breakdown of fats, proteins, and carbohydrates. Large intestine. This section absorbs water and electrolytes, transforming liquid chyme into semi-solid feces. It also houses a diverse microbiota that plays a role in fermenting undigested carbohydrates and synthesizing certain vitamins. Accessory organs. Liver produces bile, essential for emulsifying fats, and plays a significant role in metabolism and detoxification. Pancreas secretes a variety of digestive enzymes into the small intestine and regulates blood glucose levels through insulin and glucagon production. Gallbladder stores and concentrates bile, releasing it into the small intestine in response to fatty meals. Comparative digestive physiology. Different animal species exhibit variations in their digestive systems, reflecting their dietary habits. Ruminants. Animals like cattle and sheep possess a four-chambered stomach, rumen, reticulum, omasum, and abomasum, allowing them to efficiently digest fibrous plant material through microbial fermentation. Extension. MS state. Adu. Monogastric animals. Species such as humans and pigs have a single-chambered stomach. Their digestion relies heavily on enzymatic processes, making them less efficient at digesting high-fiber diets compared to ruminants. Previous year question. Question. Describe the structural and functional adaptations of the ruminant digestive system that facilitate the digestion of fibrous plant materials. Answer. Ruminants have evolved a specialized stomach divided into 4 compartments. Rumen serves as a fermentation vat where a diverse microbial population breaks down complex plant polysaccharides into volatile fatty acids, which are then absorbed as a primary energy source. Reticulum works in conjunction with the rumen to mix and regurgitate ingested material, forming cud that the animal rechews to reduce particle size and enhance microbial action. Omasum absorbs water and nutrients from the digesta, reducing its volume before it enters the abomasum. Abomasum, the true gastric stomach where acidic digestion occurs, breaking down microbial proteins and dietary proteins for absorption in the small intestine. These adaptations enable ruminants to extract maximum nutrients from fibrous plant materials, allowing them to thrive on diets predominantly composed of roughage. Multiple choice questions. Which organ is primarily responsible for the emulsification of fats during digestion? A. Stomach. B. Small intestine. C. Liver. D. Pancreas. Answer. C. Liver. In ruminants, which compartment of the stomach is known as the true stomach? A. Rumen. B. Reticulum. C. Omasum. D. Abomasum. Answer. D. Abomasum. The primary site for nutrient absorption in the human digestive system is the A. Stomach. B. Small intestine. C. Large intestine. D. Esophagus. Answer. B. Small intestine. Understanding the intricacies of the digestive system, including its comparative physiology across species, is crucial for advanced studies in animal physiology and for success in examinations like the University of Calicut PhD entrance examination in zoology. Sources. You said. Absorption. ChatGPT said. Absorption in the digestive system. Absorption is the process by which digested nutrients, water, and electrolytes pass from the lumen of the gastrointestinal tract into the bloodstream or lymphatic system. This crucial physiological function ensures that the body receives the necessary nutrients for metabolism, growth, and energy production. Absorption primarily occurs in the small intestine, although some absorption also takes place in the stomach and large intestine. The efficiency of this process depends on various structural adaptations, including villi, microvilli, and specialized transport mechanisms. Absorption in the stomach. Although the stomach is mainly responsible for mechanical and chemical digestion, it also plays a minor role in absorption. Some substances, such as water, alcohol, and certain drugs e.g., aspirin can diffuse through the stomach lining and enter the bloodstream. However, the stomach's thick mucosal lining and acidic environment limit the absorption of most nutrients. Absorption in the small intestine. The small intestine is the principal site of nutrient absorption and is divided into 3 segments: duodenum, jejunum, and ileum. Each section plays a specific role in absorbing different nutrients. Duodenum. This region receives chyme from the stomach along with bile and pancreatic enzymes. It is mainly responsible for the absorption of iron, calcium, magnesium, and some water-soluble vitamins. Jejunum. The middle section of the small intestine is highly specialized for nutrient absorption due to its dense network of villi and microvilli. Most carbohydrates, proteins, and lipids are absorbed here. Ileum. This section completes the absorption process, particularly for vitamin B12, bile salts, and fat-soluble vitamins A, D, E, and K. The ileum also contains Peyer's patches, which play a role in immune function. Structural adaptations for absorption. The small intestine has several adaptations that maximize the surface area for efficient absorption. Villi, finger-like projections that increase the surface area for nutrient absorption. Each villus contains a network of capillaries and a central lacteal for nutrient transport. Microvilli, tiny extensions of the epithelial cells lining the villi, forming a brush border that further enhances the absorptive surface. Intestinal folds, lacuae circulares. These circular folds slow down chyme movement, allowing more time for nutrient absorption. Mechanisms of nutrient absorption. Nutrient absorption occurs through various mechanisms, depending on the nature of the molecules being absorbed. Passive diffusion. Small, lipid-soluble molecules, e.g., fatty acids, move across the cell membrane without requiring energy. Facilitated diffusion. Larger molecules like fructose pass through protein channels in the membrane. Active transport. Nutrients such as glucose, amino acids, and certain ions, e.g., sodium and calcium, are transported against their concentration gradients using ATP. Endocytosis. Some large molecules, like immunoglobulins in breast milk, are absorbed through vesicular transport. Absorption of specific nutrients. Carbohydrates, broken down into monosaccharides, glucose, fructose, and galactose, which are absorbed via active or facilitated transport into the bloodstream. Proteins, absorbed as amino acids and small peptides through active transport mechanisms. Lipids, fatty acids and monoglycerides formed by cells with bile salts, allowing them to diffuse into intestinal cells, where they are reassembled into triglycerides and transported via chylomicrons into the lymphatic system. Vitamins and minerals. Water-soluble vitamins B complex, C, are absorbed through active transport or facilitated diffusion. Fat-soluble vitamins A, D, E, K require bile salts for absorption. Calcium and iron absorption is regulated by vitamin D and hepcidin, respectively. Absorption in the large intestine. The large intestine colon primarily absorbs water and electrolytes, playing a crucial role in maintaining fluid balance. It also absorbs certain vitamins, such as vitamin K and B vitamins, produced by gut microbiota. The remaining undigested material is compacted into feces for excretion. Previous year question. Question: explain the structural and functional adaptations of the small intestine to facilitate efficient nutrient absorption. Answer: the small intestine is structurally specialized for maximum nutrient absorption through the following adaptations. Villi and microvilli. These structures significantly increase the surface area of the intestinal lining, enhancing nutrient absorption. Rich capillary network. Each hose contains blood capillaries and a lacteal, ensuring efficient transport of absorbed nutrients into the bloodstream or lymphatic system. Enzymatic action. Brush border enzymes aid in the final breakdown of macromolecules before absorption. Transport mechanisms. Different molecules are absorbed via passive diffusion, facilitated diffusion, active transport, and endocytosis, ensuring efficient nutrient uptake. These adaptations enable the small intestine to absorb 90% of all digested nutrients, making it the primary site of absorption in the human body. Multiple choice questions MCQs. Which part of the digestive system is the principal site for nutrient absorption? A. Stomach. B. Duodenum. C. Small intestine. D. Large intestine. Answer: C. Small intestine. Which of the following vitamins requires intrinsic factor for absorption? A. Vitamin A. B. Vitamin B12. C. Vitamin C. D. Vitamin K. Answer: B. Vitamin B12. What is the primary function of bile in fat absorption? A. Hydrolyzing triglycerides into fatty acids. B. Transporting fatty acids into the bloodstream. C. Emulsifying fats to form micelles. D. Breaking down cholesterol. Answer: C. Emulsifying fats to form micelles. This detailed lecture provides an in-depth understanding of absorption in the digestive system, covering structural adaptations, mechanisms, and comparative physiology. These notes, along with solved previous year questions and MCQs, will be crucial for your University of Calicut PhD entrance examination in zoology preparation. Let me know the next topic. You said, energy balance. ChatGPT said. Energy balance in animal physiology. Energy balance is the state in which energy intake matches energy expenditure, ensuring that an organism maintains its body weight and physiological functions. It is a crucial aspect of animal physiology, influencing metabolism, thermoregulation, growth, reproduction, and survival. Maintaining energy balance requires precise coordination between nutrient intake, energy storage, and energy utilization. Components of energy balance. Energy balance is determined by two major factors. Energy intake. This refers to the energy obtained from food, measured in kilocalories kcal or joules J. The primary macronutrients that provide energy are carbohydrates, 4 kilocalories per gram; proteins, 4 kilocalories per gram; fats, 9 kilocalories per gram. Alcohol in some species, including humans, 7 kilocalories per gram. Energy expenditure. The body utilizes energy for various physiological functions, which can be divided into three categories. Basal metabolic rate (BMR), the minimum energy required to maintain essential physiological functions like breathing, circulation, and cellular metabolism. BMR accounts for 60–75% of total energy expenditure. Physical activity, the energy required for movement and muscular work. This varies based on activity level and species behavior. Thermogenesis, the energy used to maintain body temperature, including obligatory thermogenesis due to digestion and metabolism, and adaptive thermogenesis regulating heat production in response to environmental conditions, like shivering and cold temperatures. When energy intake equals energy expenditure, the body remains in energy balance. However, deviations from this balance can lead to energy surplus or deficit. Positive versus negative energy balance. Positive energy balance, energy intake and GT, energy expenditure. Excess energy is stored as glycogen in the liver and muscles or as fat in adipose tissue. Leads to weight gain and, in extreme cases, obesity. In animals, this state is often observed before hibernation or migration, where energy reserves are accumulated. Negative energy balance, energy intake and LT, energy expenditure. Body compensates by breaking down stored glycogen, fat, and eventually proteins for energy. Leads to weight loss, muscle wasting, and, in severe cases, metabolic disorders. Common during starvation, prolonged fasting, and extreme physical exertion. Energy balance is regulated through neuroendocrine mechanisms, primarily involving the hypothalamus, hormones, and metabolic pathways. Regulation of energy balance. Role of the hypothalamus. The hypothalamus acts as the control center for energy balance. It contains two main regions. Lateral hypothalamus, hunger center, stimulates appetite and increases food intake. Ventromedial hypothalamus, satiety center, suppresses appetite when energy stores are sufficient. Hormonal regulation of energy homeostasis. Several hormones influence hunger, satiety, and metabolism. Leptin, secreted by adipose tissue, signals satiety and decreases food intake. Ghrelin, secreted by the stomach, stimulates hunger and food intake. Insulin, regulates glucose uptake and promotes energy storage. Glucagon, stimulates glycogen breakdown and increases blood glucose. Thyroid hormones T3 and T4 increase metabolic rate and energy expenditure. Cortisol, mobilizes energy during stress by breaking down fat and protein. Disruptions in hormonal signaling can lead to metabolic disorders like obesity, diabetes, and metabolic syndrome. Energy storage and utilization. Short-term energy storage, glycogen stored in the liver and muscle serves as an immediate energy source. Glycogenolysis breaks down glycogen into glucose when energy is needed. Long-term energy storage, excess energy is stored as triglycerides in adipose tissue, which can be broken down into fatty acids during prolonged fasting or starvation. Protein catabolism, when carbohydrate and fat reserves are depleted, the body breaks down muscle proteins into amino acids, which are converted into glucose via gluconeogenesis. Energy balance in different animal groups. Different species exhibit unique adaptations for energy balance based on their environment and lifestyle. Endotherms, warm-blooded animals, maintain a constant body temperature and have higher metabolic rates. They require continuous energy intake and exhibit thermoregulation strategies such as shivering, sweating, or hibernation. Ectotherms, cold-blooded animals, depend on external temperatures to regulate metabolism. Their energy demands are lower, and they can survive extended periods without food, e.g., reptiles. Hibernation and torpor. Many animals, such as bears and rodents, enter a state of hibernation during winter, reducing metabolic rate and conserving energy. Some small mammals and birds undergo daily torpor, a short-term reduction in body temperature and metabolism to save energy. Previous year question. Question. Explain the role of hormones in regulating energy balance and metabolism in vertebrates. Answer. Energy balance and metabolism in vertebrates are regulated by multiple hormones that coordinate food intake, storage, and utilization of energy. Leptin, secreted by adipose tissue, acts on the hypothalamus to inhibit appetite and increase energy expenditure. Ghrelin, produced in the stomach, stimulates hunger and food consumption. Insulin, secreted by the pancreas, promotes glucose uptake by cells and converts excess glucose into glycogen and fat for storage. Glucagon, also from the pancreas, triggers glycogenolysis and gluconeogenesis to maintain blood glucose levels during fasting. Thyroid hormones T3 and T4 increase the basal metabolic rate, enhancing energy utilization. Cortisol, a stress hormone, mobilizes stored energy by stimulating lipolysis and protein catabolism. These hormonal interactions ensure that energy intake and expenditure are balanced, maintaining homeostasis in the body. Multiple choice questions MCQs. Which of the following hormones promotes satiety and reduces food intake? A. Ghrelin. B. Insulin. C. Leptin. D. Glucagon. Answer. C. Leptin. Energy balance and question. Explain the role of hormones in regulating energy balance and metabolism in vertebrates. Answer. Energy balance and metabolism in vertebrates are regulated by multiple hormones that coordinate food intake, storage, and utilization of energy. Leptin, secreted by adipose tissue, acts on the hypothalamus to inhibit appetite and increase energy expenditure. Ghrelin, produced in the stomach, stimulates hunger and food consumption. Insulin, secreted by the pancreas, promotes glucose uptake by cells and converts excess glucose into glycogen and fat for storage. Glucagon, also from the pancreas, triggers glycogenolysis and gluconeogenesis to maintain blood glucose levels during fasting. Thyroid hormones T3 and T4. Insulin, secreted by the pancreas. Energy balance and metabolism in vertebrates are regulated by multiple hormones that coordinate food intake, storage, and utilization of energy. Leptin, secreted by adipose tissue, acts on the hypothalamus to inhibit appetite and increase energy expenditure. Ghrelin, produced in the stomach, stimulates hunger and food consumption. Insulin, secreted by the pancreas, promotes glucose uptake by cells and converts excess glucose into glycogen and fat for storage. Glucagon, also from the pancreas, triggers glycogenolysis and gluconeogenesis to maintain blood glucose levels during fasting. Thyroid hormones T3 and T4 increase the basal metabolic rate, enhancing energy utilization. Cortisol, a stress hormone, mobilizes stored energy by stimulating lipolysis and protein catabolism. These hormonal interactions ensure that energy intake and expenditure are balanced, maintaining homeostasis in the body. Multiple choice questions MCQs. Which of the following hormones promotes satiety and reduces food intake? A. Ghrelin. B. Insulin. C. Leptin. D. Glucagon. Answer. C. Leptin. Which hormone stimulates glycogen breakdown to maintain blood glucose levels? A. Insulin. B. Glucagon. C. Leptin. D. Cortisol. Answer. B. Glucagon. Which of the following is the primary long-term energy storage molecule in animals? A. Glycogen. B. ATP. C. Triglycerides. D. Proteins. Answer. C. Triglycerides. This detailed lecture on energy balance covers all key aspects necessary for your University of Calicut PhD entrance examination in zoology, including comprehensive theory, previous year solved questions, and MCQs. Let me know the next topic. You said, BMR. ChatGPT said, basal metabolic rate BMR introduction. Basal metabolic rate BMR is the minimum amount of energy required by an organism to maintain vital physiological functions while at complete rest. These functions include breathing, circulation, cell maintenance, thermoregulation, and brain activity. BMR is expressed in kilocalories kcal per day or joules per hour per kilogram of body weight. It represents 60-75% of total daily energy expenditure and is a critical factor in energy balance, metabolism, and body weight regulation. Factors affecting BMR. Several physiological and environmental factors influence BMR, including body size and composition. Lean body mass. Muscle tissue is metabolically active and requires more energy than fat tissue, meaning individuals with higher muscle mass have a higher BMR. Fat tissue. Adipose tissue has a lower metabolic activity, so individuals with higher fat percentage have a lower BMR. Age. BMR declines with age due to the loss of muscle mass and reduction in metabolic activity. Infants and children have a higher BMR per unit body weight due to rapid growth and higher energy demands. Sex. Males generally have a higher BMR than females because of greater muscle mass and lower body fat percentage. Females have a lower BMR, primarily due to higher fat storage and hormonal differences. Genetics. Genetic factors influence metabolic rate, body composition, and hormonal regulation, affecting BMR variability among individuals. Hormonal regulation. Thyroid hormones T3 and T4, thyroxine T4, and triiodothyronine T3 play a crucial role in increasing metabolic rate. Hyperthyroidism elevates BMR, while hypothyroidism reduces it. Growth hormone GH stimulates protein synthesis and increases BMR. Cortisol increases energy mobilization that can suppress BMR in chronic stress conditions. Insulin plays a role in glucose metabolism and energy storage, indirectly influencing BMR. Body temperature and climate. Higher body temperature, fever, warm climates increases metabolic reactions, leading to a higher BMR. Cold environments trigger thermogenesis, increasing BMR to maintain body heat. Diet and nutritional status. Starvation or severe caloric restriction lowers BMR as a survival mechanism. Frequent meals and protein-rich diets slightly increase BMR due to the thermic effect of food. Physical activity. Regular exercise increases muscle mass, leading to a higher resting metabolic rate, RMR, which is closely related to BMR. Sedentary lifestyle reduces muscle mass, leading to a lower BMR. Measurement of BMR. BMR is measured under strict resting conditions to eliminate external influences. The standard conditions for BMR measurement include Complete rest lying down but awake post-absorptive state 12-14 hours after the last meal, neutral temperature environment, thermoneutrality, no physical or emotional stress methods of measuring BMR direct calorimetry. Measures the heat produced by the body in a controlled chamber. Highly accurate but expensive and not commonly used. Indirect calorimetry. Measures oxygen consumption (FO) and carbon dioxide production (BCO) to estimate energy expenditure. The respiratory quotient (RQ) ratio of CO produced to O consumed helps determine the predominant metabolic fuel, carbohydrates, fats, or proteins. Predictive equations. Harris-Benedict. While BMR represents the baseline energy requirement, the total energy expenditure T consists of basal metabolic rate BMR 60-75% energy needed for vital functions. Physical activity 15-30% energy used for voluntary movements. Thermic effect of food TEF 10% energy required for digestion, absorption, and metabolism of food. When energy intake matches T, body weight remains stable. An imbalance leads to weight gain positive energy balance or weight loss negative energy balance. BMR and clinical significance obesity and BMR. Obese individuals often have a lower BMR per unit body weight due to reduced muscle mass relative to fat mass. Weight loss strategies focus on increasing BMR through exercise and muscle building activities. Starvation and BMR. Prolonged fasting or severe calorie restriction lowers BMR as the body conserves energy. This metabolic adaptation makes long-term weight loss difficult. Hyperthyroidism and BMR. Excess thyroid hormone production leads to increased BMR, causing weight loss, excessive sweating, and hyperactivity. Hypothyroidism and BMR. Deficiency of thyroid hormones leads to reduced BMR, resulting in weight gain, fatigue, and slow metabolism. Aging and BMR. BMR naturally declines with age, requiring adjustments in diet and activity levels to maintain a healthy energy balance. Previous year question. Question. Discuss the physiological factors that regulate basal metabolic rate BMR in mammals. Answer. Basal metabolic rate BMR is regulated by multiple physiological factors, including body composition. Higher muscle mass increases BMR, whereas higher fat percentage decreases it. Age and sex. BMR decreases with age due to muscle loss. Males typically have a higher BMR than females. Hormonal influence. Thyroid hormones T3 and T4 are the primary regulators, with hyperthyroidism increasing BMR and hypothyroidism decreasing it. Genetics. Inherited metabolic traits influence individual variations in BMR. Environmental factors. Cold environments increase BMR due to thermogenesis, while warm environments may reduce it. Nutritional status. Starvation reduces BMR, whereas protein-rich diets can slightly enhance it. BMR is a critical determinant of total energy expenditure, and its regulation is essential for maintaining metabolic homeostasis in mammals. Multiple choice questions MCQs. Which of the following hormones has the greatest impact on BMR? A. Insulin. B. Cortisol. Answer. C. Thyroxine T4. BMR is typically highest in which of the following individuals? A. An elderly woman with high body fat. B. A young male athlete with high muscle mass. C. A person with hypothyroidism. D. A person in prolonged fasting. Answer. B. A young male athlete with high muscle mass. Which method is commonly used to estimate BMR? A. Direct calorimetry. B. Indirect calorimetry. C. Genetic testing. D. X-ray imaging. Answer. B. Indirect calorimetry. This comprehensive lecture on basal metabolic rate BMR covers all essential aspects for the University of Calicut PhD entrance examination in zoology, including detailed theory, solved previous year questions, and MCQs. Let me know the next topic. You said. Blood and circulation. ChatGPT said. Blood and circulation introduction. Blood and circulation are essential components of the cardiovascular system, responsible for transporting oxygen, nutrients, hormones, and waste products throughout the body. The circulatory system consists of the heart, blood vessels, and blood. Blood circulation ensures the maintenance of homeostasis, thermoregulation, immune defense, and gas exchange. Blood, composition and functions. Composition of blood. Blood is a connective tissue composed of plasma 55% and formed elements 45%, including red blood cells RBCs, white blood cells WBCs, and platelets. 1. Plasma. Plasma is the fluid component of blood, constituting 55% of total blood volume. It is composed of 90% water, 7% plasma proteins, albumin, globulins, fibrinogen, and 3% dissolved substances, nutrients, hormones, gases, waste products. Functions of plasma. Maintains blood volume and pH, transports nutrients, hormones, and waste products, regulates body temperature. 2. Formed elements of red blood cells, erythrocytes. RBCs are biconcave, anucleate cells containing hemoglobin for oxygen transport. Produced in the bone marrow via erythropoiesis, stimulated by the hormone erythropoietin EPO. Lifespan, 120 days, after which they are destroyed in the spleen and liver. Functions. Oxygen transport from lungs to tissues. Carbon dioxide transport from tissues to lungs. B. White blood cells, leukocytes. WBCs are nucleated cells responsible for immune defense. Types of WBCs. Granulocytes, neutrophils, eosinophils, basophils, agranulocytes, lymphocytes, monocytes. Functions. Neutrophils, phagocytosis of pathogens. Eosinophils, defense against parasites, allergy response. Basophils, histamine release in allergic reactions. Lymphocytes. WBCs are nucleated cells responsible for immune defense. Types of WBCs. Granulocytes, neutrophils, eosinophils, basophils, agranulocytes, lymphocytes, monocytes. Functions. Neutrophils, phagocytosis of pathogens. Eosinophils, defense against parasites, allergy response. Basophils, histamine release in allergic reactions. Lymphocytes, antibody production. B cells and cell-mediated immunity. T cells. Monocytes, become macrophages, engulf pathogens. C. Platelets, thrombocytes. Small, anucleate cell fragments involved in blood clotting hemostasis. Produced in the bone marrow from megakaryocytes. Release clotting factors like thrombin and fibrin to form a clot. Circulatory system. The circulatory system consists of pulmonary circulation, blood flow between the heart and lungs. Systemic circulation, blood flow between the heart and body tissues. Coronary circulation, blood supply to the heart muscle itself. Types of circulatory systems. Open circulatory system, found in arthropods and mollusks. Blood flows into open spaces, sinuses, instead of enclosed vessels. Closed circulatory system, found in vertebrates. Blood flows through a network of blood vessels with the heart as a pumping organ. Heart, structure and function. The heart is a muscular organ that functions as a pump to circulate blood. It consists of 4 chambers. 2 atria, right and left, receive blood. 2 ventricles, right and left, pump blood out. Circulatory pathway. Right atrium, right pointing arrow, right ventricle, right pointing arrow, pulmonary artery, right pointing arrow, lungs, oxygenation, pulmonary vein, right pointing arrow, left atrium, right pointing arrow, left ventricle, right pointing arrow, aorta, right pointing arrow, body. The heart functions through contractions regulated by electrical impulses generated by the sinoatrial node, natural pacemaker. Blood vessels. Blood vessels transport blood throughout the body and are classified as arteries, thick-walled vessels that carry oxygenated blood away from the heart except pulmonary artery. Veins, thin-walled vessels that carry deoxygenated blood toward the heart except pulmonary vein. Capillaries, thin, single-cell thick vessels that facilitate exchange of gases, nutrients, and waste. Blood pressure and its regulation. Blood pressure BP is the force exerted by blood against vessel walls. It is measured as systolic BP SBP, pressure during ventricular contraction, approximately 120 mm of mercury. Diastolic BP DBP, pressure during ventricular relaxation, approximately 80 mm of mercury. Regulation of BP. Neural control. Medulla oblongata regulates BP via the autonomic nervous system. Hormonal control. Renin-angiotensin-aldosterone system ROS increases BP. Antidiuretic hormone ADH increases water retention, raising BP. Atrial natriuretic peptide ANP decreases BP by promoting sodium excretion. Cardiac cycle and heart sounds. The cardiac cycle is a sequence of events in one heartbeat, lasting 0.8 seconds, and includes atrial systole, 0.1 seconds, atria contract, pushing blood into ventricles. Ventricular systole, 0.3 seconds, ventricles contract, ejecting blood into arteries. Diastole, 0.4 seconds, heart relaxes, allowing blood to fill chambers. Heart sounds. Lub S1, closing of atrioventricular AV valves, mitral and tricuspid. Dub S2, closing of semilunar valves, aortic and pulmonary. Disorders of blood and circulation. Hypertension, high BP, persistent high BP increases heart disease risk. Atherosclerosis, plaque buildup in arteries, leading to reduced blood flow. Anemia, low RBC counter hemoglobin, causing oxygen deficiency. Leukemia, cancer of WBCs, leading to immune dysfunction. Thrombosis, formation of blood clots in vessels, causing blockages. Myocardial infarction, heart attack, blockage of coronary arteries leading to heart tissue damage. Previous year question. Question, explain the mechanisms of blood pressure regulation and its physiological importance. Answer. Blood pressure is regulated by a combination of neural, hormonal, and renal mechanisms to ensure adequate tissue perfusion. Neural regulation, the medulla oblongata regulates BP via the autonomic nervous system. The sympathetic nervous system SNS increases BP by vasoconstriction, while the parasympathetic nervous system PNS lowers BP by vasodilation. Hormonal regulation. Renin-angiotensin-aldosterone system ROS increases BP by vasoconstriction and sodium retention. Antidiuretic hormone ADH increases BP by water retention. Atrial natriuretic peptide ANP lowers BP by promoting sodium excretion. Renal regulation, the kidneys regulate BP by controlling blood volume through sodium and water balance. Maintaining BP is essential for adequate tissue oxygenation, nutrient transport, and waste removal. Dysregulation can lead to conditions like hypertension and hypotension. Multiple choice questions MCQs, which of the following is the main pacemaker of the heart? A. AV node. B. Purkinje fiber. C. SAW node. D. Bundle of his answer. C. SAW node. Which blood vessel carries oxygenated blood from the lungs to the heart? A. Pulmonary vein. B. Pulmonary artery. C. Aorta. D. Superior vena cava. Answer. A. Pulmonary vein. Which hormone lowers blood pressure by promoting sodium excretion? A. ADH. B. Aldosterone. C. Renin. D. ANP. Answer. D. ANP. This comprehensive lecture covers blood and circulation for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Blood corpuscles and formed elements. ChatGPT said. Blood corpuscles and formed elements. Introduction. Blood is a specialized connective tissue that performs vital functions such as oxygen transport, immune defense, and coagulation. It consists of plasma 55% and formed elements 45%. The formed elements, also known as blood corpuscles, include red blood cells, erythrocytes, white blood cells, leukocytes, and platelets, thrombocytes. These cellular components originate from hematopoietic stem cells in the bone marrow through a process called hematopoiesis. Red blood cells, erythrocytes, structure and morphology. RBCs are biconcave, disc-shaped, anucleate cells, allowing maximum surface area to volume ratio for efficient gas exchange. Their flexibility enables them to pass through narrow capillaries. They contain hemoglobin, a metalloprotein responsible for oxygen binding and transport. Function of RBCs. Oxygen transport. Hemoglobin binds oxygen in the lungs and releases it into tissues. Carbon dioxide transport. RBCs carry CO as bicarbonate ions, HCO, to the lungs for exhalation. pH regulation. The bicarbonate buffer system maintains blood pH homeostasis. Production and life cycle. Erythropoiesis. The formation of RBCs occurs in the bone marrow under the stimulation of erythropoietin, EPO, a hormone secreted by the kidneys in response to low oxygen levels, hypoxia. Lifespan. RBCs survive for 120 days, after which they are phagocytosed by macrophages in the spleen, liver, and bone marrow. Breakdown. Hemoglobin is degraded into hemoglobin. Heme is converted to bilirubin, excreted in bile, while iron is recycled for new RBC synthesis. Disorders of RBCs. Anemia, a condition with low RBC counter hemoglobin levels, leading to fatigue and weakness. Polycythemia, excess RBC production, increasing blood viscosity and the risk of clots. Sickle cell disease, a genetic disorder where RBCs become crescent-shaped, causing blockages in capillaries. White blood cells, leukocytes. Classification of WBCs. White blood cells are nucleated, motile cells involved in immune defense. They are classified into granulocytes and agranulocytes based on the presence of cytoplasmic granules. Granulocytes neutrophils 50-70%. Phagocytic cells that engulf pathogens and cellular debris. Contain lysosomal enzymes and play a key role in innate immunity. First responders to infections and inflammation. Eosinophils 2-4%. Defend against parasitic infections, e.g., helminths. Involved in allergic reactions by releasing histaminase, which counteracts histamine from basophils. Basophils and LT, 1%. Secrete histamine and heparin, mediating inflammatory responses. Play a role in anaphylactic reactions and hypersensitivity responses. Agranulocytes. Lymphocytes 20-40%. B lymphocytes. B cells produce antibodies for humoral immunity. T lymphocytes. T cells mediate cell-mediated immunity. Natural killer NK cells destroy virus-infected and cancerous cells. Monocytes 2-8%. Differentiate into macrophages in tissues, which phagocytose pathogens in dead cells. Present antigens to lymphocytes, activating adaptive immune responses. Functions of WBCs. Neutrophils and monocytes, phagocytosis of pathogens. Lymphocytes, antibody production and adaptive immunity. Eosinophils, defense against parasites and allergic response. Basophils, release histamine, triggering inflammation. Disorders of WBCs. Leukopenia, low WBC count, increasing infection susceptibility. Leukocytosis, elevated WBC count, often due to infections or leukemia. Leukemia, a type of blood cancer characterized by uncontrolled WBC proliferation. Platelets, thrombocytes. Structure and origin. Platelets are anucleate, disc-shaped cell fragments derived from megakaryocytes in the bone marrow. They circulate in the blood and function in hemostasis, blood clotting. Function of platelets. Primary hemostasis. Platelets adhere to damaged blood vessel walls, forming a temporary plug at the site of injury. They release clotting factors like ADP, thromboxane A, and serotonin, promoting further aggregation. Secondary hemostasis. The clotting cascade activates fibrin, which stabilizes the platelet plug and forms a blood clot. This process prevents excessive blood loss and initiates wound healing. Coagulation cascade. The coagulation cascade consists of intrinsic pathway, initiated by damaged endothelial surfaces. Extrinsic pathway, triggered by tissue injury and exposure to tissue factor. Common pathway, leads to conversion of prothrombin to thrombin, which converts fibrinogen to fibrin, forming a stable clot. Disorders of platelets. Thrombocytopenia, low platelet count, leading to excessive bleeding. Thrombocytosis, high platelet count, increasing the risk of blood clots. Hemophilia, a genetic disorder where clotting factors are deficient, causing prolonged bleeding. Previous year question. Question, describe the role of erythropoietin in erythropoiesis and its physiological significance. Answer. Erythropoietin EPO is a glycoprotein hormone primarily produced by the kidneys in response to hypoxia, low oxygen levels. It plays a crucial role in stimulating erythropoiesis, RBC production, in the bone marrow. Mechanism of action of erythropoietin. Detection of hypoxia, the kidneys sense low oxygen levels and release EPO into the bloodstream. Stimulation of bone marrow, EPO binds to receptors on erythroid progenitor cells, promoting RBC maturation and proliferation. Increased oxygen transport, newly formed RBCs increase oxygen delivery to tissues, restoring homeostasis. Physiological importance of erythropoietin. Maintains RBC homeostasis. Enhances oxygen carrying capacity, prevents anemia in chronic diseases e.g., kidney failure. Used therapeutically in anemia treatment and is a performance-enhancing drug in endurance sports. Multiple choice questions, MCQs. Which of the following WBCs is responsible for antibody production? A. Neutrophils. B lymphocytes. C monocytes. D. Physiological importance of erythropoietin. Maintains RBC homeostasis. Enhances oxygen carrying capacity, prevents anemia in chronic diseases e.g., kidney failure. Used therapeutically in anemia treatment and is a performance-enhancing drug in endurance sports. Multiple choice questions, MCQs. Which of the following WBCs is responsible for antibody production? A. Neutrophils. B lymphocytes. C monocytes. D basophils. Answer, B lymphocytes. Which blood cell plays a major role in allergic reactions? A. Neutrophils. B monocytes. C basophils. D erythrocytes. Answer, C basophils. Which organ is the primary site of erythropoietin production? A. Liver. B spleen. C kidneys. D bone marrow. Answer, C kidneys. This completes the detailed lecture on blood corpuscles and formed elements for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said, plasma function. ChatGPT said, plasma and its functions. Introduction. Blood plasma is the fluid component of blood, making up about 55% of total blood volume. It is a pale yellow, slightly alkaline fluid that serves as a transport medium for various substances. Plasma consists of 90-92% water, 7-8% plasma proteins, and 1-2% other solutes such as nutrients, gases, electrolytes, and waste products. It plays a crucial role in maintaining homeostasis, transport, immunity, and blood clotting. Composition of plasma. 1. Water 90-92%. Acts as a solvent for dissolved substances. Helps maintain blood volume and pressure. Regulates temperature by distributing heat throughout the body. 2. Plasma proteins 7-8%. Plasma proteins are produced mainly by the liver and perform various physiological functions. Albumins 55-60%. Maintain colloid osmotic pressure, preventing excessive water loss from blood vessels. Transport hormones, drugs, and fatty acids. Globulins 35-40%. Alpha and beta globulins transport lipids, vitamins, and hormones. Gamma globulins, immunoglobulins, antibodies are essential for immune defense. Fibrinogen 4-6%. Plays a key role in blood clotting coagulation by converting into fibrin threads to form a clot. Prothrombin. A precursor to thrombin, essential for blood coagulation. 3. Electrolytes, ions. Plasma contains sodium, not potassium, K, calcium, Ca², chloride, Cl, and bicarbonate, HCO, ions, which help in maintaining osmotic balance and acid-base equilibrium, regulating nerve and muscle function. 4. Gases. Oxygen, O, dissolved in plasma, but most is carried by hemoglobin and RBCs. Carbon dioxide, CO, transported as bicarbonate ions, HCO, dissolved CO, and carbaminohemoglobin. Nitrogen, N, inert gas with no direct physiological function. 5. Nutrients and metabolic waste. Plasma transports glucose, amino acids, fatty acids, vitamins, and minerals required for cellular metabolism. It carries waste products like urea, creatinine, ammonia, and uric acid to excretory organs. Functions of plasma. 1. Transport function. Nutrients, carries glucose, amino acids, fatty acids, and vitamins from the digestive system to tissues. Gases, oxygen and carbon dioxide are transported between the lungs and tissues. Hormones, plasma carries hormones secreted by endocrine glands to target organs. Metabolic waste, removes nitrogenous wastes like urea, creatinine, and ammonia to the kidneys for excretion. 2. Maintenance of blood volume and osmotic balance. Albumins maintain oncotic pressure, preventing excessive fluid loss from blood vessels. Electrolytes like sodium and chloride help regulate water balance and blood pressure. 3. Immunity and defense mechanism. Immunoglobulins, antibodies, neutralize pathogens. Complement proteins help destroy bacteria and infected cells. 4. Blood clotting and wound healing. Plasma proteins fibrinogen and prothrombin are essential for coagulation. Prevents excessive bleeding during injury. 5. Acid-base balance, pH regulation. Plasma contains buffer systems like the bicarbonate buffer HCO to maintain a stable pH of 7.35-7.45. 6. Heat regulation. Plasma distributes heat from metabolically active organs, liver, muscles to other parts of the body, maintaining temperature homeostasis. Previous year question. Question. Explain the role of plasma proteins in blood homeostasis. Answer. Plasma proteins play a vital role in maintaining homeostasis by performing various physiological functions. Regulation of osmotic pressure. Albumin maintains oncotic pressure, preventing excessive fluid loss from capillaries. Ensures proper circulation and blood volume balance. Transport function. Albumin and globulins transport hormones, lipids, and drugs. Help in the distribution of nutrients, gases, and metabolic waste. Immune defense. Gamma globulins, immunoglobulins neutralize pathogens, playing a key role in adaptive immunity. Complement proteins assist in bacterial lysis and immune response activation. Blood clotting. Fibrinogen and prothrombin help in coagulation, preventing excessive bleeding during injury. Fibrin stabilizes blood clots and promotes wound healing. Acid-base balance. Plasma proteins act as buffers, stabilizing blood pH by binding or releasing hydrogen ions H. Heat regulation. Plasma absorbs, distributes, and regulates body temperature, ensuring thermal homeostasis. Multiple choice questions, MCQs. Which plasma protein is primarily responsible for maintaining osmotic pressure? A. Globulin. B. Fibrinogen. C. Albumin. D. Prothrombin. Answer. C. Albumin. Which plasma protein is involved in blood clotting? A. Immunoglobulin. B. Fibrinogen. C. Albumin. D. Myoglobin. Answer. B. Fibrinogen. What is the primary function of gamma globulins in plasma? A. Oxygen transport. B. Immune defense. C. Clot formation. D. Blood buffering. Answer. B. Immune defense. This detailed lecture on plasma and its functions covers everything you need for the University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said, blood. Volume, blood volume regulation, blood groups, ChatGPT said. Blood volume regulation, and blood groups blood volume. Definition and normal range. Blood volume refers to the total amount of blood circulating in the body. In an average adult. Males, 5 to 6 liters. Females, 4 to 5 liters. The volume of blood is approximately 7-8% of total body weight and consists of 55% plasma and 45% formed elements, RBCs, WBCs, and platelets. Blood volume is crucial for circulatory stability, oxygen transport, and homeostasis. Blood volume regulation. The regulation of blood volume is essential for maintaining blood pressure, tissue perfusion, and fluid balance. Involves a dynamic balance between fluid intake, distribution, and excretion, regulated by the kidneys, cardiovascular system, endocrine system, and autonomic nervous system. 1. Role of the kidneys. The kidneys regulate blood volume through glomerular filtration rate, GFR, controls how much plasma is filtered. Renin-angiotensin-aldosterone system, ROS, increases blood volume by promoting sodium and water retention. Antidiuretic hormone, ADH, promotes water reabsorption in the kidneys, reducing urine output and increasing blood volume. 2. Role of the cardiovascular system. Baroreceptors in the aorta and carotid arteries detect changes in blood pressure and send signals to regulate heart rate and blood vessel diameter. Vasoconstriction, sympathetic stimulation, increases blood pressure, while vasodilation, parasympathetic activity, decreases it. 3. Role of the endocrine system. Aldosterone from adrenal glands increases sodium and water retention, expanding blood volume. Atrial natriuretic peptide, ANP, secreted by the heart when blood volume is high, promoting sodium excretion and reducing blood volume. 4. Fluid balance and osmoregulation. Hypothalamus detects changes in plasma osmolality and stimulates thirst, leading to water intake. Excess water intake reduces blood osmolarity, leading to increased urine production. 5. Hemorrhage and blood loss compensation. During hemorrhage, blood volume drops, activating sympathetic responses and hormonal mechanisms to restore it. Capillary fluid shifts help redistribute extracellular fluids into circulation. Blood groups. Discovery and importance. Blood groups were first discovered by Carl Landsteiner in 1901, leading to the ABO blood group system. Blood groups are determined by the presence or absence of specific antigens on the surface of RBCs and their corresponding antibodies in plasma. ABO blood group system. The ABO system is based on 2 antigens A and B on RBC membranes and 2 antibodies NEA and anti-B in plasma. Blood group antigens on RBCs. Antibodies in plasma can receive blood from can donate to AA anti-BA, OA, AB. BB anti-AB, OB, AB. AB and B non-A, B, AB, O, universal recipient, ABO non-anti-A and anti-BOA, B, AB, O, universal donor, universal donor. The Rh factor, rhesus factor, is another important antigen present plus or absent minus on RBCs. If Rh positive, Rh, the person has the Rh antigen. The Rh factor, rhesus factor, is another important antigen present plus or absent minus on RBCs. If Rh positive, Rh, the person has the Rh antigen. If Rh negative, Rh, the antigen is absent. An Rh negative mother carrying an Rh positive fetus may develop anti-Rh antibodies, leading to hemolytic disease of the newborn HDN in subsequent pregnancies. Other blood group systems. Apart from ABO and Rh, other systems include Kell system, important in transfusion reactions. Duffy system, linked to malaria resistance. Lewis and Kidd systems, play roles in organ transplantation and transfusion compatibility. Previous year question. Question. Describe the mechanisms involved in the regulation of blood volume. Answer. Blood volume regulation is a complex process involving multiple physiological mechanisms to maintain circulatory stability. The key mechanisms include Renin-angiotensin-aldosterone system, ROS. Renin, released by the kidneys in response to low blood pressure, converts angiotensinogen to angiotensin I, which is further converted into angiotensin II by the angiotensin-converting enzyme ACE. Angiotensin II causes vasoconstriction, increasing blood pressure and stimulating the release of aldosterone, which promotes sodium and water retention, expanding blood volume. Antidiuretic hormone, ADH. Secreted by the hypothalamus and released from the posterior pituitary in response to high plasma osmolality. Increases water reabsorption in the kidneys, reducing urine output and restoring blood volume. Atrial natriuretic peptide, ANP. Secreted by the heart atria when blood volume is excessive. Promotes sodium excretion, natriuresis, and diuresis, lowering blood volume. Sympathetic nervous system, SNS, regulation. Baroreceptors in the aortic arch and carotid arteries detect blood pressure changes. Sympathetic activation causes vasoconstriction and increased heart rate, helping maintain blood pressure and volume. Capillary fluid shifts. During blood loss or dehydration, fluid from the interstitial space moves into circulation to compensate. When blood volume is high, excess fluid moves into the interstitial tissues. Thirst mechanism. Osmoreceptors in the hypothalamus detect dehydration and stimulate thirst, promoting fluid intake. These mechanisms work in coordination to ensure stable blood volume, preventing hypovolemia, low blood volume, and hypervolemia, excess blood volume. Multiple choice questions, MCQs. Which hormone plays a key role in increasing blood volume by promoting sodium and water retention? A. Insulin. B. Aldosterone. C. Thyroxine. D. Glucagon. Answer. B. Aldosterone. Which of the following blood groups is considered a universal donor? A positive. B. AB negative. C. O negative. D. B positive. Answer. C. O negative. Which organ is responsible for producing plasma proteins involved in blood clotting? A. Kidneys. B. Liver. C. Spleen. D. Pancreas. Answer. B. Liver. This comprehensive lecture on blood volume regulation, and blood groups is tailored for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Hemoglobin, immunity, homeostasis, ChatGPT said. Hemoglobin, immunity, and homeostasis. Hemoglobin. Introduction. Hemoglobin HB is an iron-containing oxygen transport metalloprotein found in red blood cells, RBCs. It plays a crucial role in oxygen transport, carbon dioxide removal, and acid-base balance. Each hemoglobin molecule consists of 4 polypeptide chains, globins, and 4 heme groups, with each heme containing an iron base-squared ion that binds oxygen. Structure of hemoglobin. Hemoglobin is a tetrameric protein composed of 2 alpha-alpha and 2 beta-beta globin chains in adults, HbA. Each globin chain contains a heme group with ferrous iron base-squared which can bind 1 oxygen molecule. 1 hemoglobin molecule can carry 4 oxygen molecules. O. Functions of hemoglobin. 1. Oxygen transport. Hemoglobin binds oxygen in the lungs, high oxygen tension, forming oxyhemoglobin. It releases oxygen in tissues, low oxygen tension, where it is needed for cellular respiration. 2. Carbon dioxide transport. About 20% of CO is carried as carbaminohemoglobin bound to hemoglobin. CO is also transported as bicarbonate ions, HCO, in plasma. 3. Acid-base balance buffering action. Hemoglobin acts as a buffer by maintaining blood pH through the Bohr effect. Oxygen release is enhanced in acidic conditions. 4. Regulation of nitric oxide, NO, levels. Hemoglobin binds and releases nitric oxide, NO, which regulates blood vessel dilation and improves blood flow. Types of hemoglobin. Adult hemoglobin, HbA, the most common form in adults, alpha-beta. Fetal hemoglobin, HbF, found in fetuses, alpha-gamma, has a higher oxygen affinity for better oxygen uptake from the mother's blood. Hemoglobin A, HbA, a minor adult form, alpha-delta. Abnormal hemoglobins. Sickle hemoglobin, HbS, causes sickle cell anemia. HbC, HbE, found in genetic hemoglobinopathies. Hemoglobin disorders. Anemia, low hemoglobin levels due to iron deficiency, blood loss, or bone marrow disorders. Polycythemia, excessive hemoglobin production, increasing blood viscosity. Thalassemia, genetic disorder causing defective hemoglobin synthesis. Sickle cell disease, mutation in the beta-globin gene causes sickle-shaped RBCs, leading to blockages in blood flow. Immunity. Introduction. Immunity is the body's defense mechanism against infections, foreign substances, and abnormal cells. It is classified into innate immunity, nonspecific, and adaptive immunity, specific. 1. Innate immunity, nonspecific immunity. Innate immunity is present from birth and provides a rapid, general response to pathogens. Components of innate immunity. Physical barriers, skin, mucous membranes, tears, and saliva. Cellular defense, macrophages, neutrophils, natural killer NK cells, dendritic cells. Chemical defense. Lysozyme in tears and saliva destroys bacterial cell walls. Interferons inhibit viral replication. Complement system enhances phagocytosis and inflammation. 2. Adaptive immunity, specific immunity. Adaptive immunity develops in response to specific pathogens and involves memory cells for long-term protection. It is mediated by lymphocytes B and T cells. Types of adaptive immunity: humoral immunity, B cells and antibodies. B cells produce antibodies, immunoglobulins, that neutralize pathogens. Types of antibodies: Ig, IgA, IgM, IgE, and IgD. Cell-mediated immunity, T cells. Helper T cells, CD4, activate B cells and macrophages. Cytotoxic T cells, CD8, kill virus-infected and cancer cells. Regulatory T cells, suppress excessive immune responses. 3. Types of immunity. Natural immunity, acquired through infection or maternal antibodies. Artificial immunity, acquired through vaccination or antibody injections. Homeostasis. Introduction. Homeostasis is the process of maintaining a stable internal environment despite external fluctuations. It is essential for survival, cellular function, and physiological balance. Mechanisms of homeostasis. 1. Negative feedback mechanisms. Most homeostatic processes operate via negative feedback loops, where a change triggers responses that restore balance. Examples. Blood glucose regulation, insulin lowers blood sugar, glucagon raises it. Body temperature regulation, sweating cools the body, shivering generates heat. Blood pressure control, baroreceptors detect changes and adjust heart rate. 2. Positive feedback mechanisms. Positive feedback amplifies a response until a specific event occurs. Examples. Childbirth, oxytocin release, contractions intensify until delivery. Blood clotting, platelets release signals to form a clot. Previous year question. Question. Describe the structure and functions of hemoglobin in detail. Answer. Hemoglobin is a tetrameric protein composed of 2 alpha and 2 beta globin chains, each containing a heme group with base squared. It performs the following functions. Oxygen transport. Binds oxygen in the lungs and releases it in tissues. The Bohr effect enhances oxygen unloading in acidic environments. Carbon dioxide transport. Transports CO as carbaminohemoglobin and bicarbonate ions. Acid-base balance. Acts as a buffer, stabilizing blood pH. Regulation of nitric oxide, no. Controls blood vessel dilation. Hemoglobin disorders like sickle cell anemia, thalassemia, and anemia affect oxygen transport and overall health. Multiple choice questions MCQs. Which metal ion is present in the heme group of hemoglobin? A. Calcium, Ca squared. B. Magnesium, Mg squared, C. Iron, Fe squared, D. Copper, Cu squared. Answer, C. Iron, Fe squared. Which immunoglobulin is primarily responsible for allergic reactions? A. Ig. B. IgA, C. IgE, D. IgM. Answer, C. IgE. Which organ is the primary regulator of homeostasis? A. Heart. B. Brain, hypothalamus, C. Lungs. D. Pancreas. Answer, B. Brain, hypothalamus. This comprehensive lecture on hemoglobin, immunity, and homeostasis is tailored for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Comparative anatomy of heart structure, myogenic heart, ChatGPT said. Comparative anatomy of heart structure and myogenic heart introduction. The heart is a vital organ responsible for pumping blood throughout the body. Across the animal kingdom, heart structure and function vary significantly depending on the circulatory system type, open or closed, and the metabolic needs of the organism. The hearts of different vertebrates show evolutionary modifications that enhance circulatory efficiency, while invertebrate hearts exhibit similar yet effective designs. Comparative anatomy of heart structure 1. Invertebrate hearts. Protozoa, lack a true heart, circulation occurs through cytoplasmic streaming or diffusion. Porifera, sponges, no specialized circulatory system, water flows through canals. Cnidarians, jellyfish, corals utilize a gastrovascular cavity for circulation. Annelids, earthworms, have contractile aortic arches, hearts, that pump blood. Arthropods, insects, crustaceans, possess a dorsal tubular heart with ostia openings. Blood is pumped into the open circulatory system, bathing tissues directly. Mollusks, snails, octopuses. Most have chambered hearts with atria and ventricles. Cephalopods, squids, octopuses have a closed circulatory system with multiple hearts, 1 systemic, 2 bronchial. 2. Vertebrate hearts from fishes to mammals. Vertebrate hearts evolved from simple 2-chambered structures in fish to complex 4-chambered hearts in mammals and birds. A fish heart, 2-chambered. Single circulation, blood flows once through the heart per cycle, composed of 1 atrium and 1 ventricle. Deoxygenated blood is pumped to the gills for oxygenation and then distributed to the body. B. Amphibian heart, 3-chambered. Double circulation begins, but with some mixing of oxygenated and deoxygenated blood. Consists of 2 atria and 1 ventricle. Oxygenated blood from the lungs and deoxygenated blood from the body mix in a single ventricle. C. Reptilian heart, partially 4-chambered. Most reptiles except crocodiles have incomplete ventricular separation, reducing blood mixing. The crocodilian heart is fully divided, allowing complete separation of oxygenated and deoxygenated blood. D. Bird and mammalian heart, 4-chambered. Complete separation of oxygenated and deoxygenated blood. Ensures efficient double circulation with a high metabolic rate. Left side pumps oxygenated blood to the body, systemic circulation, while the right side pumps deoxygenated blood to the lungs, pulmonary circulation. Myogenic heart. Definition. A myogenic heart is a heart where contractions originate from the cardiac muscle itself, rather than external nervous stimulation. The heartbeats are initiated by pacemaker cells within the myocardium, making the heart capable of autonomous function. Characteristics of myogenic hearts. Found in vertebrates e.g., mammals, birds, reptiles, amphibians, fish. The sinoatrial SA node acts as the natural pacemaker. Electrical impulses travel through the atrioventricular AV node, bundle of His, and Purkinje fibers, ensuring coordinated contraction. Regulated by the autonomic nervous system, but does not rely on nerve impulses to initiate beats. Neurogenic versus myogenic hearts. Feature neurogenic heart, myogenic heart. Found in arthropods e.g., insects, crustaceans, vertebrates e.g., humans, birds, pacemaker located in a nerve ganglion located in the heart muscle. You said, no table ChatGPT said. Neurogenic versus myogenic hearts. A neurogenic heart is one where the heartbeat originates from nerve impulses rather than the heart muscle itself. This type is found in arthropods such as insects and crustaceans. In these organisms, a nerve ganglion regulates the heartbeat, and if the nerves are cut, the heart stops beating. In contrast, a myogenic heart generates its own impulses within the cardiac muscle. This type is found in vertebrates, including fish, amphibians, reptiles, birds, and mammals. The heartbeat originates from specialized pacemaker cells in the sinoatrial SA node. The impulse then spreads through the atrioventricular AV node, bundle of His, and Purkinje fibers, ensuring coordinated contraction of the heart chambers. Since the heart generates its own rhythm, it continues beating even if disconnected from the nervous system. However, autonomic nerves can modulate the heart rate, increasing it via sympathetic stimulation or decreasing it via parasympathetic control. Previous year question. Question. Explain the evolutionary transition of heart structure in vertebrates and the significance of a 4-chambered heart in mammals and birds. Answer. The vertebrate heart has evolved from a simple 2-chambered structure in fish to a complex 4-chambered heart in mammals and birds, ensuring greater circulatory efficiency. In fishes, the heart consists of 1 atrium and 1 ventricle, functioning in a single circulatory system where deoxygenated blood is pumped to the gills for oxygenation. In amphibians, the heart has 2 atria and 1 ventricle, introducing double circulation but with some mixing of oxygenated and deoxygenated blood. In reptiles, the ventricle is partially divided, reducing the mixing of oxygenated and deoxygenated blood. In birds and mammals, the heart is fully divided into 4 chambers, completely separating oxygenated and deoxygenated blood. This results in efficient oxygen transport, high metabolic rates, and sustained activity levels, which is particularly crucial for endotherms that require continuous energy production. Multiple choice questions MCQs. 1. In which group of animals does a 2-chambered heart function in a single circulatory system? A. Amphibians. B. Birds. C. Mammals. D. Fish. Answer, D. Fish. 2. What is the primary pacemaker of a myogenic heart? A. Purkinje fibers. B. Atrioventricular AV node. C. Sinoatrial SA node. D. Bundle of His. Answer, C. Sinoatrial SA node. 3. In which of the following animals is a neurogenic heart found? A. Frogs. B. Crustaceans. C. Mammals. D. Reptiles. Answer, B. Crustaceans. This detailed lecture on comparative anatomy of the heart and myogenic hearts is structured for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Specialized tissue, ECG, its principle and significance ChatGPT said. Specialized cardiac tissue, ECG, principle and significance introduction. The heart's ability to contract rhythmically and pump blood efficiently is due to the presence of specialized cardiac tissue. This tissue consists of pacemaker cells and conduction fibers that generate and propagate electrical impulses, ensuring synchronized contraction of the heart chambers. The electrocardiogram ECG records these electrical activities, providing crucial insights into heart function and diagnosing cardiac disorders. Specialized cardiac tissue. Types of specialized tissue in the heart. The heart contains autorhythmic cells that generate impulses and contractile cells that respond to these signals. The key components of a specialized cardiac conduction system are sinoatrial SAW node, the natural pacemaker of the heart, located in the right atrium. It initiates the heartbeat by generating electrical impulses at regular intervals. Atrioventricular AV node, located at the junction of the atria and ventricles. It delays the impulse slightly before allowing it to pass into the ventricles, ensuring proper atrial contraction before ventricular contraction. Bundle of His, a group of specialized fibers that transmit impulses from the AV node to the ventricles. It divides into the right and left bundle branches. Purkinje fibers, these are the terminal branches of the conduction system, spreading electrical impulses throughout the ventricles, ensuring a rapid and synchronized contraction. The coordination between these components ensures that the heart beats rhythmically and efficiently pumps blood throughout the body. Electrocardiogram ECG, principle and significance. Principle of ECG. The electrocardiogram ECG or EKG is a diagnostic tool that records the electrical activity of the heart over time. It is based on the principle that electrical impulses generated by the heart can be detected and recorded through electrodes placed on the skin. These impulses create a pattern of electrical changes, which are displayed as waves on an ECG graph. The ECG measures the potential difference between 2 points on the body, which corresponds to the depolarization and repolarization of cardiac muscles. ECG waves and their significance. P wave represents atrial depolarization, which leads to atrial contraction. Any abnormality in this wave may indicate atrial dysfunction. QRS complex represents ventricular depolarization, which leads to ventricular contraction. A prolonged or altered QRS complex can indicate ventricular conduction abnormalities. T wave represents ventricular repolarization, the recovery phase before the next heartbeat. T wave abnormalities can suggest electrolyte imbalances or ischemia. PR interval, the time between the onset of atrial depolarization and ventricular depolarization. A prolonged PR interval may indicate a heart block. ST segment represents the plateau phase of ventricular depolarization. Changes in the ST segment, such as elevation or depression, are crucial indicators of myocardial infarction, heart attack, or ischemia. Significance of ECG in diagnosis. Detect arrhythmias, irregular heart rhythms, such as tachycardia, fast heart rate, or bradycardia, slow heart rate, can be diagnosed using ECG. Identifying myocardial infarction, changes in the ST segment and the appearance of abnormal Q waves help in diagnosing heart attacks. Monitoring heart function, ECG is used in routine checkups and for monitoring patients with heart diseases. Detecting conduction disorders, abnormalities in the PR interval or QRS complex can indicate conduction system defects such as heart blocks. Effects of drugs and electrolyte imbalances, ECG helps assess the impact of medications and electrolyte disturbances, e.g., hyperkalemia, hypokalemia, on heart function. Previous year question. Question, explain the principle of ECG and its significance in diagnosing cardiac disorders. Answer. The electrocardiogram ECG records the electrical activity of the heart by detecting voltage changes that occur during the cardiac cycle. It is based on the principle that the heart generates electrical impulses that can be recorded through electrodes placed on the skin. These impulses create characteristic waveforms, including the P wave, QRS complex, and T wave, which correspond to different phases of cardiac activity. ECG is clinically significant for detecting various heart conditions. Arrhythmias such as atrial fibrillation and ventricular tachycardia can be diagnosed based on irregular ECG waveforms. Myocardial infarction can be identified by ST segment changes in the presence of abnormal Q waves. ECG also helps detect heart blocks, electrolyte imbalances, and the effects of medications on cardiac function. By analyzing ECG patterns, physicians can evaluate overall cardiac health and make timely interventions in case of abnormalities. Multiple choice questions, MCQs. 1. Which part of the cardiac conduction system is known as the natural pacemaker? A. Atrioventricular AV node. B. Sinoatrial SAW node. C. Bundle of His. D. Purkinje fibers. Answer. B. Sinoatrial SAW node. 2. The QRS complex in an ECG represents A. Atrial depolarization. B. Ventricular depolarization. C. Ventricular repolarization. D. Atrial repolarization. Answer. B. Ventricular depolarization. 3. Which of the following changes in an ECG is most commonly associated with myocardial infarction? A. Prolonged PR interval. B. ST segment elevation. C. Tall P waves. D. Inverted T waves. Answer. B. ST segment elevation. This comprehensive lecture on specialized cardiac tissue and ECG is tailored for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Cardiac cycle, heart as a pump, blood pressure, neural and chemical regulation of all above. ChatGPT said. Cardiac cycle, heart as a pump, blood pressure, and neural and chemical regulation. Introduction. The heart functions as a highly efficient pump that maintains continuous blood circulation to supply oxygen and nutrients to tissues. The cardiac cycle describes the sequence of events in a single heartbeat, while blood pressure represents the force exerted by circulating blood on vessel walls. The heart's activity is regulated by neural and chemical mechanisms to ensure proper function under different physiological conditions. Cardiac cycle. The cardiac cycle refers to the complete sequence of events during one heartbeat, involving contraction, systole, and relaxation, diastole, of the heart chambers. It lasts about 0.8 seconds in a healthy human at rest and consists of 3 main phases. Atrial systole, 0.1 seconds, the atria contract, pushing blood into the relaxed ventricles. The atrioventricular AV valves, tricuspid and bicuspid, mitral valves remain open, while the semilunar valves, aortic and pulmonary valves remain closed. Ventricular systole, 0.3 seconds, the ventricles contract, increasing pressure and forcing blood into the aorta and pulmonary artery. The AV valves close to prevent backflow, and the semilunar valves open to allow blood ejection. Diastole, 0.4 seconds, the heart muscles relax, allowing blood to flow into the atria from the veins. The semilunar valves close to prevent backflow, while the AV valves reopen, preparing for the next cycle. The lub-dub sound of the heartbeat is produced by the closure of the heart valves. The first sound S1 due to AV valve closure and the second sound S2 due to semilunar valve closure. Heart as a pump. The heart operates as a dual pump, maintaining two distinct circulatory pathways. Pulmonary circulation, right side of the heart, pumps deoxygenated blood to the lungs for oxygenation. Systemic circulation, left side of the heart, pumps oxygenated blood to the rest of the body. The pumping efficiency of the heart is determined by cardiac output, CO, the amount of blood pumped by each ventricle per minute. It is calculated as CO equals heart rate, HR, times stroke volume, SV, where HR is the number of beats per minute, and SV is the volume of blood ejected per beat. Stroke volume, SV, the amount of blood ejected by each ventricle per contraction, about 70 milliliters in a healthy adult. Ejection fraction, EF, the percentage of blood ejected from the ventricles per beat, indicating heart efficiency. A normal EF is 55-70%. Frank-Starling mechanism, increased venous return stretches the ventricular muscle, leading to a stronger contraction and higher stroke volume, ensuring efficient circulation. Blood pressure. Blood pressure, BP, is the force exerted by circulating blood against the walls of arteries. It is measured in millimeters of mercury, mmHg, and recorded as systolic pressure, diastolic pressure, e.g., 120/80ths of a millimeter of mercury. Systolic blood pressure, SVP, the pressure during ventricular contraction, systole, when blood is ejected into the arteries. Diastolic blood pressure, DVP, the pressure during ventricular relaxation, diastole, when the heart refills with blood. Factors affecting blood pressure. Cardiac output, higher CO increases BP. Peripheral resistance, narrower arteries increase BP due to higher resistance. Blood volume, more blood increases BP. Blood viscosity, thicker blood increases BP. Elasticity of arteries, reduced elasticity, as in arteriosclerosis, raises BP. Neural and chemical regulation of the cardiac cycle, heart pumping, and blood pressure neural regulation. The autonomic nervous system, ANS, plays a crucial role in heart function and BP regulation. Sympathetic nervous system, SNS, increases heart rate, tachycardia, force of contraction, and BP by releasing norepinephrine. Parasympathetic nervous system, PNS, decreases heart rate, bradycardia, and BP via the vagus nerve, releasing acetylcholine. Baroreceptors, pressure sensors located in the aortic arch and carotid sinuses, they detect BP changes and send signals to the brainstem, adjusting heart rate and vessel dilation accordingly. Chemoreceptors, chemical sensors located in the carotid and aortic bodies, they respond to oxygen, carbon dioxide, and pH levels in the blood, adjusting respiration and cardiac function as needed. Chemical regulation. Several hormones influence heart function and BP. Adrenaline, epinephrine, and noradrenaline, norepinephrine, secreted by the adrenal medulla, they increase heart rate, contractility, and BP. Renin-angiotensin-aldosterone system, ROS, helps regulate blood volume and BP. Renin from kidneys activates angiotensin II, which constricts blood vessels and stimulates aldosterone, leading to sodium and water retention, increasing BP. Antidiuretic hormone, ADH, vasopressin, released by the hypothalamus, it increases water retention and blood volume, raising BP. Atrial natriuretic peptide, ANP, secreted by the heart in response to high BP, it promotes sodium excretion and vasodilation, reducing BP. Previous year question. Question, explain the role of neural and chemical mechanisms in the regulation of blood pressure. Answer. Blood pressure regulation involves neural and chemical mechanisms to maintain homeostasis. The autonomic nervous system, ANS, regulates heart rate and vessel diameter. The sympathetic nervous system releases norepinephrine, increasing heart rate and vasoconstriction, thereby raising BP. The parasympathetic nervous system, through the vagus nerve, releases acetylcholine, slowing heart rate and reducing BP. Baroreceptors in the aortic arch and carotid sinuses detect BP fluctuations and adjust cardiac output and vascular tone accordingly. Chemoreceptors in the carotid and aortic bodies monitor oxygen and CO levels, adjusting respiratory and circulatory functions. Chemical regulation involves hormones such as adrenaline, which increases BP, and renin-angiotensin-aldosterone system, ROS, which enhances vasoconstriction and fluid retention. Antidiuretic hormone, ADH, promotes water retention, increasing BP, whereas atrial natriuretic peptide, ANP, lowers BP by reducing blood volume. Together, these mechanisms maintain stable blood pressure essential for organ perfusion. Multiple choice questions, MCQs. 1. During which phase of the cardiac cycle do the ventricles contract and eject blood? A. Atrial systole. Ventricular systole. C. Ventricular diastole. D. Atrial diastole. Answer. B. Ventricular systole. 2. Which of the following hormones increases blood pressure by promoting sodium retention? A. Atrial natriuretic peptide, ANP. B. Insulin. C. Aldosterone. D. Oxytocin. Answer. C. Aldosterone. 3. The baroreceptors that help regulate blood pressure are located in the lungs and liver. B. Aortic arch and carotid sinuses. C. Pancreas and kidneys. D. Small intestine and spleen. Answer. B. Aortic arch and carotid sinuses. This detailed lecture on the cardiac cycle, heart pumping, blood pressure, and its regulation is structured for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Respiratory system, comparison of respiration in different species, ChatGPT said. Respiratory system, comparison of respiration in different species. Introduction. Respiration is a fundamental biological process that enables organisms to obtain oxygen for cellular metabolism and expel carbon dioxide, a byproduct of energy production. Different species have evolved specialized respiratory structures adapted to their environment, metabolic demands, and evolutionary lineage. Respiration can occur through diffusion, gills, tracheae, lungs, or specialized respiratory pigments that enhance oxygen transport. The efficiency of respiration depends on factors such as surface area, diffusion gradients, and respiratory pigments. Respiration in different groups of organisms. 1. Protozoans and simple invertebrates, amoeba, paramecium, sponges, cnidarians, flatworms. These organisms lack specialized respiratory organs and rely entirely on simple diffusion across their body surface. Oxygen diffuses into the body from the surrounding environment, and carbon dioxide is expelled in the opposite direction. The process is efficient only in small, thin-bodied organisms with a high surface area to volume ratio. 2. Aquatic invertebrates, annelids, mollusks, arthropods, echinoderms. Many aquatic invertebrates possess gills for respiration. Annelids, earthworms, marine worms use cutaneous respiration, where oxygen diffuses across the moist body surface. Some marine species have gills. Mollusks, snails, clams, octopuses. Gills are present in aquatic forms, while terrestrial gastropods, land snails, have a vascularized mantle cavity functioning as a primitive lung. Arthropods, crustaceans, insects, arachnids. Crustaceans use gills, while insects use tracheal systems and network of air tubes. Arachnids, spiders, scorpions have book lungs or tracheae. Echinoderms, starfish, sea urchins use dermal branchiae, skin gills, and tube feet for gas exchange. 3. Aquatic vertebrates, fish and amphibians. Fish respiration. Fish use gills for respiration, extracting oxygen from water through countercurrent exchange. Oxygen-rich water flows over gill filaments in the opposite direction of blood flow, maximizing oxygen uptake. Some fish, like lungfish, can use a primitive lung in low-oxygen environments. Amphibian respiration. Amphibians exhibit dual respiration, using gills, lungs, and skin at different life stages. Larvae tadpoles have external gills. Adult amphibians, frogs, salamanders use lungs but also rely on cutaneous respiration, skin breathing in moist conditions. Some salamanders are lungless and rely entirely on cutaneous respiration. 4. Terrestrial vertebrates, reptiles, birds, mammals, reptile respiration. Reptiles have more efficient lungs than amphibians but lack a diaphragm. They rely on negative pressure ventilation, where muscles expand the rib cage to draw in air. Some species, like sea turtles, have specialized cloacal respiration, extracting oxygen through vascularized tissue in the cloaca. Bird respiration. Birds have the most efficient respiratory system among vertebrates. They possess air sacs and parabronchial lungs, allowing unidirectional airflow. This system ensures continuous oxygen supply even during exhalation, supporting the high metabolic demand of flight. The cross-current exchange system in bird lungs maximizes oxygen absorption, making it superior to mammalian lungs. Mammal respiration. Mammals rely entirely on lungs, which are ventilated by the diaphragm and intercostal muscles. The lungs contain alveoli, which provide a large surface area for gas exchange. Oxygen is transported via hemoglobin in red blood cells, and respiration is regulated by chemoreceptors detecting blood CO levels. Adaptations in extreme environments. High-altitude animals, yak, llama, snow leopard, bar-headed goose, have higher lung capacity, increased red blood cell count, and hemoglobin with high oxygen affinity to survive in low oxygen conditions. Diving mammals, seals, whales, dolphins store oxygen in myoglobin-rich muscles, have collapsible lungs to avoid decompression sickness, and reduce heart rate bradycardia during deep dives. Desert animals, kangaroo rat, camel minimize water loss by excreting concentrated urine and extracting metabolic water from food. Previous year question. Question. Compare the respiratory adaptations in fish and birds and explain how these adaptations support their respective lifestyles. Answer. Fish and birds have evolved specialized respiratory systems suited to their environments. Fish rely on gills, which are highly vascularized and use a countercurrent exchange mechanism to maximize oxygen absorption from water. Since water has lower oxygen content than air, this adaptation ensures efficient extraction of oxygen. In contrast, birds have unidirectional airflow in their lungs, supported by air sacs that allow continuous oxygen supply even during exhalation. The cross-current exchange in bird lungs ensures higher oxygen uptake, which is essential for high metabolic rates required for flight. This respiratory efficiency allows birds to fly at high altitudes where oxygen levels are low. Thus, fish are adapted to aquatic respiration using gills, while birds possess the most efficient respiratory system among vertebrates, supporting their active and energy-demanding lifestyle. Multiple choice questions, MCQs. 1. Which of the following organisms rely on cutaneous respiration as their primary mode of gas exchange? A. Birds. B. Mammals. C. Amphibians. D. Reptiles. Answer. C. Amphibians. 2. What is the key feature of avian respiration that makes it more efficient than mammalian respiration? A. Diaphragm-assisted ventilation. B. Unidirectional airflow through lungs. C. Presence of alveoli. D. Use of book lungs. Answer. B. Unidirectional airflow through lungs. 3. In fish, the countercurrent exchange mechanism functions to A. Minimize oxygen absorption. B. Maximize oxygen uptake from water. C. Reduce metabolic rate. D. Increase carbon dioxide retention. Answer. B. Maximize oxygen uptake from water. This detailed lecture on comparative respiration is structured for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said, anatomical considerations, transport of gases, exchange of gases, waste elimination. ChatGPT said. Respiratory, anatomical considerations, gas transport, gas exchange, and waste elimination. Introduction. The respiratory system is essential for oxygen intake, carbon dioxide removal, and maintaining homeostasis. In different species, respiration varies based on anatomical structures adapted to specific environments. This topic explores the anatomy of respiratory structures, transport of gases in the body, gas exchange at the tissue and cellular levels, and the elimination of gaseous waste. Anatomical considerations of the respiratory system. The anatomy of the respiratory system varies among different organisms based on their evolutionary adaptations. 1. Invertebrate respiratory anatomy. Protozoans, sponges, and cnidarians rely on direct diffusion through the body surface due to their simple body structure. Annelids use cutaneous respiration, where gases diffuse through a moist body surface. Some species have external gills. Mollusks, aquatic forms have gills, while terrestrial gastropods have a vascularized mantle cavity functioning as a lung. Arthropods, insects use tracheal systems with spiracles and branching tubes, while crustaceans have gills. Echinoderms use dermal branchiae, skin gills, and tube feet for respiration. 2. Vertebrate respiratory anatomy. Fish have gills with filaments and lamellae, enabling oxygen extraction from water using a countercurrent exchange system. Amphibians show dual respiration using gills in larva, lungs in adults, and cutaneous respiration. Reptiles use lungs with increased surface area for efficient gas exchange. Birds have a unique air sac system allowing unidirectional airflow, maximizing oxygen absorption. Mammals possess alveoli-rich lungs, where oxygen diffuses into the bloodstream and carbon dioxide is expelled. Transport of gases. 1. Oxygen transport. Oxygen is transported via the respiratory pigments present in the blood. In vertebrates, oxygen binds to hemoglobin in red blood cells, forming oxyhemoglobin (HbO) in the lungs or gills. In invertebrates, respiratory pigments include hemocyanin in mollusks and arthropods, hemerythrin in some annelids, and chlorocruorin in marine polychaetes. Oxygen transport is influenced by partial pressure, PO temperature, pH Bohr effect, and carbon dioxide levels. 2. Carbon dioxide transport. Carbon dioxide is transported in three main forms: dissolved CO, 7-10% directly dissolved in plasma. Carbaminohemoglobin, 20-30%, CO binds to hemoglobin. Bicarbonate ions, HCO, 60-70%, CO reacts with water to form carbonic acid, HCO, which dissociates into bicarbonate and hydrogen ions. The enzyme carbonic anhydrase catalyzes this reaction, maintaining acid-base balance. Exchange of gases. 1. Gas exchange at the lungs or gills, external respiration. Oxygen enters the blood. Oxygen from air or water diffuses across the respiratory membrane, alveolar epithelium in mammals, gill filaments in fish, into the blood. Carbon dioxide exits the blood. CO diffuses from the blood into the lungs or water in aquatic organisms and is exhaled. 2. Gas exchange at tissues, internal